have an amine group, then what makes the difference between the 20 of them? And we will discuss that. Primarily, it's there's a side group, but therefore if there are 20 different amino acids that are responsible for biological synthesis, that are necessary for biological synthesis, then that means that every protein in your body, everything from your hair to the crystalline in your eyes, to the hemoglobin that carries oxygen, to the proteins that are cross-linked to make your little toenail, are all made out of the exact same 20 amino acids. This is one of the reasons why it doesn't matter if you're eating beans or steak. As long as there's protein there, you are getting the amino acid components that you need in order to build you. This is yet another example of how when they say you are what you eat, this is not a euphemism, it is quite literally the truth, because the molecules that you get from the food that you eat are digested down into their individual chemical components, which your body then uses to synthesize the stuff that makes you you. Yes?
When I was in high school, years ago, they were telling us that there were 21 different amino acids that were essential.
Well, there are two problems with essays.
What got demoted?
There are two problems with essays. One, 20, not 21. And I don't know which one that they might have been talking about that could have been demoted in terms of what you're discussing. And two, there are not 20 essential amino acids. There are 20 amino acids that are relevant for biological synthesis. There are less than that are essential amino acids, and there are many, many more than that you can actually find in the proteins that you have, but all of them are post-translational modifications. And so you start with something after you build the protein, then it comes on and it modifies this amino acid with this extra functional group, or cuts that functional group off of that amino acid, and all of those differences have new names after they are modified. So there are 20 that we need to make it, but we end up with a lot more than 20 when we're done, and fewer than 20 of those are essential, which we will have a slide about, but essential meaning the same thing as when we were talking about fatty acids. Essential means what? What do we call an essential fatty acid? Where do you get an essential fatty acid and why? We just talked about this on Tuesday. Essential means that you cannot synthesize them. Your body does not know how to make them. And therefore you have to get them from the things that you eat. There was a relatively short list of essential fatty acids that you had to get from your diet. There is a relatively short list of essential amino acids that you have to get from your diet. This is one of the reasons why, when I was a freshman in college, I had some friends and knew of even of other people as well that wanted to try out the whole vegetarian thing, just didn't want to eat meat anymore. And there are two ways to do that. There's the right way, and there's the way that most people do it, which aka the wrong way, which is just to stop eating meat. Well, there's a problem with that. Meat has a relatively good, different animals have different ratios, but meat has a relatively good balance of all the essential amino acids that you need. If you're going to cut that out of your diet, then you have to make sure that what you are eating gives you enough of all of those things. And most people, when they decide to just become a vegetarian, they just decide to stop eating meat, and they replace that meat with something like pasta, because it's cheap and easy, and they're in college and don't want to cook really complex meals. Don't want to have to pay much attention to the details of exactly what they're eating. Being an appropriate vegetarian, a healthy vegetarian, is actually a lot of work. It's a lot easier just to cook a steak. Well, it's a lot easier just to cook a bad steak. Cooking a good steak is also a challenge, but just eating meat is a good way to get all of those amino acids that you need. And so therefore, most of the time when people decide they want to be vegetarian as a freshman in college, they gain more than the freshman 15, because frequently they don't do it right. They just stop eating meat and replace it with a lot of other unhealthy things. Like that's going to somehow magically make them healthier. It is the average or stereotypical weight gain of a freshman who is no longer being fed by their parents in school at home, but who is now responsible for their own diet and food intake, and so therefore they overeat, under-exercise, and gain about 15 pounds.
I've never heard of that before.
Yeah, it's a thing. Not everyone has it. It is a stereotype.
No one leaves freshman year without 15 pounds.
Right.
I actually lost some pounds. So.
Shush, don't want to hear it. Yeah, guess who found them?
I imagine students who go to college on a very an extremely energetic athletic scholarship. Maybe they're not like maybe they would be excluded from that. It doesn't happen to everyone, but weight gain in your freshman year is a very common thing.
Well, they make you stay in the dorms, and you have a meal plan to a buffet.
Right. Yeah.
You feel like you have to eat everything.
That's why they did away with the trays.
Oh, really?
Right now, so that's
Oh, you don't have that buffet pass?
no, well, it's not open yet.
When you take a tray, you
It
just
opens in
fill
the
so
fall
much up and then throw it all away. So they quit doing trays.
Some of these proteins function as structural components, so just like the cellulose for carbohydrates, it was a major structural component of plant cells in the form of that cell wall. We also can have major structural components that come in the form of proteins. Cartilage, muscle fibers, hair, nails, all of these guys are made out of predominantly proteins. Enzymes, as I mentioned before, they're relevant for pretty much anything that is work that is occurring within your body is responsible or carried out on the part of enzymes. There are, I think, 99.9% instead of 100%. 99.9 might be an overestimation as well, but it's something hot in the high 90s because there are a couple of other things that can also metabolize or catalyze certain reactions, ribozymes or otherwise. But most of the time, the vast majority of the time, if work is being done, and I'm talking about like pushing a fridge up a 30-degree incline type of work, building things, breaking things, that work is being done by enzymes, which are, for the most part, proteins. Hemoglobin is a protein that transports oxygen that we talked about before, and we'll talk about more today. Myoglobin is a similar oxygen transport molecule that you find within the blood and especially within your muscle cells, because muscles are going to have a large oxygen requirement. So is your brain. Your brain is the most energetically costly organ that you have individually within your body. It uses the most energy because thinking is expensive, yo. All right, here's a lovely table of some examples of some of these different classes of proteins: structural, contractile, transport, storage, collagen, myosin. Myosin and actin being the contractile aspects of the muscle fibers. Myosin and actin, the myosin molecule is literally walking, almost like a little Charlie Chaplin type of guy, walking along the actin filaments within your muscles, and that's what causes the contraction of the muscles. This is why muscles only pull, because a muscle only has force when it shortens itself. It cannot elongate itself. All it can do is relax and cause the myosin fibrils to let go of the actin fibrils, and then something else has to actually pull against it to lengthen it. A relaxed muscle is not going to just lengthen very much on its own unless something else pulls against it. And this is why our muscles always come in pairs. When you have your biceps and your triceps, your biceps pulls on the arm, and your triceps also pull on the arm, but they pull on the opposite side to elongate that particular joint, and elongate the bicep muscle that you had just used before that. Storage proteins like casein is a protein storage in milk. Ferritin is a protein that involves storing iron in the spleen and the liver to help make sure that you don't go through anemic periods. We've got enzymatic aspects, hormones. Insulin, one of the hormones that we've talked about a little bit before, is actually a protein. Enzymes, which I mentioned before, protection, in the act of recognizing and destroying foreign substances like these immunoglobulins, your immune system, both in how it recognizes red blood cells that are not supposed to be there to protect you, and also in how it recognizes foreign invaders like viruses and bacteria and amoeba and protists that are invading your system that should not be there. The way in which those immune cells recognize that foreign body, the way in which they attack that foreign body, the way in which they clear that foreign body, all of that is regulated by proteins. And all proteins are made out of amino acids. So when we talked about carbohydrates, the monomer for carbohydrate was the saccharide, the monosaccharide. And the carbohydrate, especially in the cases of cellulose or starch, was this massive, massive molecule of hundreds to thousands to tens of thousands of glucose molecules that were bound together in long or branching chains, right? Well, now when we talk about amino acids, we're talking once again about a macromolecule that can get to be huge, but it's made out of the combination of the monomers, i.e. the amino acids together, one after another, 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 anywhere between 50 to thousands of amino acids long to form the proteins that we make. So I might ask you, how is it, do you think, can you conceptualize the idea, how is it possible that given that there are only 20 different amino acids shown here in their basic structure, the ammonium group on one side, the amino group, the carboxylic acid group, or the acid part, so we have the amino acid, all of them look like this. They all have that central carbon with one hydrogen hanging off, and then the thing that is different between the 20 that are relative is this little orange group, relative, or illustrated here by a little CH3 group. We can replace that CH3 group with an ethyl group instead of that methyl group that we have there. We could add an ethyl group, we could add a propyl group, we could add an isopropyl group, we could add a histine ring structure, we could add all sorts of different things to it. There are way more amino acids than 20. There are just only 20 that are relevant for biological synthesis. So your body needs 20 of them to start this process. It could then modify them to be something else after that is done. And outside of biological synthesis, in biochemistry or organic chemistry or pseudo-organic chemistry, we could make amino acids that are exactly this, where that R group is some sort of massive convoluted structure. But when it comes to the synthesis of living cells of your body, it's going to be just those 20 that matter. So, how can you explain how you can have everything from the enzymes that allow you to think, to the enzymes that digest your food, to the enzymes, or I should say the proteins that make your hair, to the proteins that make the different color of your eyes, to the proteins that are responsible for the movement of your muscles, to the proteins that are responsible for the digestion of your food. How can you make all of those different enzymes with just 20 different amino acids? Is this a concept that is challenging to wrap your head around? I hope it's not. Because I can ask you a corollary, I can give you a metaphor. How much information could you store if I gave you a group of 20 things and asked you how much you could do with those 20 things? I would hope that you would be able to at least conceptually imagine that you could do a near limitless amount of things with those 20 things. Let's say I called that list of 20 things an alphabet. How much information could you store with that? With just 6 more letters, literally every tidbit of information that's ever been known in the English language has been stored. With just 26 letters. How did we do that? How could 26 letters store all information known to English-speaking people? I suppose it's cheating. You can say 26 letters plus 10 alphanumeric characters. But still, the concept remains the same. What's the difference between Moby Dick and the Bible?
One is true.
I was waiting for that one.
I suppose I walked into that one.
Hey, you don't know, you weren't there.
Hey, both involved a whale.
I've never seen a whale before.
Hey, it's not a whale, it's a large fish.
That's true. That is true.
Uh-huh.
It's not
If
a
we're
mammal.
going to get into arguments about semantics, about
biology.
allegory. But, what's the difference? If they're both, presuming, of course, they're both written in English, what's the difference between any two books?
Their sequencing is different.
The order of the letters, right? The same 26 letters, but just in different orders. So how could you get enzymes that can do such different things? By changing the order of the letters. Especially when you're talking about enzymes that could be as small as 50 amino acids or as large as 10,000. You can get very, very different functions. But all of them, every single one of them, started like this, with just differences in that side group right there. So here's our amine group, here's our carboxylic acid group, here's that central carbon, and then it's whatever replaces this guy right here as an R group. That's what separates one amino acid from another. Some of those amino acids are nonpolar, or otherwise known as hydrophobic, meaning they do not get along well with water, right? Amino acids with side groups that have large carbon chains, therefore that do not have things that interact well with water, are going to be hydrophobic amino acids. Now, allow me to ask you this question. Do you think that valine itself, this particular amino acid, do you think that that is soluble in water? The amino acid itself, because we are describing the amino acid as a non-polar, therefore hydrophobic amino acid. So is this amino acid, is that soluble in water?
No.
It would be tempted to say no, but the answer is of course it is. Because look at that right there. And look at that right there. We get a whole bunch of highly charged things, but here's the thing. If you take this amino acid and shove it into a huge protein, everything in blue doesn't matter anymore. The only thing that matters is the stuff in yellow hanging off the side. So when we group a bunch of these things together, if we have a bunch of these guys together, then that is going to create a region of that protein that does not get along with water. Whereas this guy right here, that side group from asparagine, will get along with water just fine. Why? Because we have our carbonyl functional group here and an amine functional group right there. Plenty of opportunities for water to hydrogen bond with it, right? But individually, all of these amino acids are soluble in water. Because when they're floating around by themselves, not part of a chain, they've got a whole bunch of polar groups on them. When we describe them as polar or non-polar, what we're describing is the nature of the side chain. When you involve these amino acids in polypeptides, in the larger synthesis, the larger molecules, when we link them together, what is the chemical nature of their side chain? Therefore, what chemical properties is that side chain going to grant to that region of the protein? That's what we're describing when we describe them as hydrophilic or hydrophobic. Not the amino acid itself, floating around in water about to be used in synthesis, but the nature of the amino acid when it is used in a protein. You probably could have even answered that question, especially in the case of valine, quite easily. Because remember, where's the cutoff for carboxylic acids? For solubility in water?
6 carbon.
So around 6 or 7 is when we start having minor problems, right? But that's just if the only thing we have is that carboxylic acid functional group right there. We've got that and the amine group hanging off the other side. So what do you think that would do? Push it even deeper? Maybe into the 8 or 9 range? An amino acid is nonpolar when the R group is either H, a bunch of alkyl groups, or aromatic groups. Because aromatic groups are made out of benzene, right? Or ring structures. And therefore they're going to have a lot of electron density that's not going to get along well with water and therefore will be nonpolar. So here we have the most basic of amino acids, very at the top right there, glycine. You'll notice that all of these guys it hasn't been given to us yet, but all of them have 3-letter abbreviations and single-letter abbreviations. Glycine is abbreviated GLY or just G. Alanine, where we replace the hydrogen with a methyl group, that was that first one we were introduced to, alanine. That is abbreviated ALA or just A. We've got valine, abbreviated VAL or V, where we replace the methyl group with an isopropyl group. Leucine, where we add a butyl group, isobutyl group. Isoleucine, where we add once again a butyl group, but it's in a different shape. Instead of 2-methylpropane, or 2-methylpropane, yes, 2-methylpropane. Instead of 2-methylpropane, it's 1-methylpropane, which should just be called butane. But you can see how these two things have different groups. Isoleucine is abbreviated ILE or just I. Not all of the first letter abbreviations for these amino acids make sense because we have 20 of them, but some of them start with the same first letter, and so we can't just use the first letter as the abbreviation for all of them. For example, down here, tryptophan is this weird or aromatic fungicide group, but the one letter abbreviation for tryptophan is W. There is a T, I believe that's tyrosine. Tyrosine tryptophan. One of them gets to be called, they don't get to be called T, both get to be called T. Right? What is it?
Threonine?
Threonine for T? There we go.
Yeah.
What's tyrosine? I used to have all this memorized. Y? There we go. So, you can see how all of these R groups here have a lack of anything that's getting along well with water, right? We have a little bit here and there. We have that nitrogen right there that you could argue, well, maybe that nitrogen could have a little bit of a charge, sure, but not so much. There's a whole bunch of electron density around it that isn't going to do much. And so here we have non-polar groups. We can call them polar when we have either a hydroxyl, a thiol that's not linked, because here we have a sulfur in methionine, but it's already bound to two different things, right? Whereas this cysteine has a thiol, true thiol group, S bonded to an H. So if we have something that is electronegative and electropositive with those partial dipoles that allow us to have a hydrogen bonding, so here we have a hydroxyl group off the end, that can hydrogen bond, another hydroxyl, a thiol group, here we have a carbonyl with an amine group, and another carbonyl with an amine group. These are both what type of bonds? What do we call the type of bonds that combines a carbonyl group with an amine group at the end? It's a special type of amine bond that we call an amide, right? An amine bond is just carbon to nitrogen. An amide bond is nitrogen to a carbon that already has a double bonded oxygen, right? So here's our list of polar amino acids. I don't expect you to memorize this, but a really easy question to ask is which of these amino acids is polar? I don't expect you to memorize it. You could if you wanted to, but you could also just do what? Remember where you can look it up. Right. It's the benefit of an online open book test, right? You can look this stuff up. Assuming, of course, you understand what I mean when I ask which of these is a polar amino acid. All of these guys are polar. Yes. All of those guys are non-polar. And on top of that, we also have polar charged amino acids, here called acidic amino acids. So these are polar negatively charged, because back here, these are polar amino acids, right? But they do not have a complete charge. That hydroxyl right there has not donated its hydrogen and gained a negative charge. It is still neutral. But the OH bond in that hydroxyl does have partial charge, right? That's what it allows it to hydrogen bond with water. The carboxylic acid group here at aspartate and glutamate at pH, at physiological pH, which is 7.4, both of those hydrogens that would have been attached to that single bonded oxygen, both of those hydrogens have been donated. And so therefore, that side group is both polar and negatively charged. So here we have aspartate and glutamate. Do you guys remember when we talked about MSG? What does MSG stand for?
Monosodium glutamate.
Monosodium glutamate, because we have two different carboxylic acid groups here that could form salts with sodium. Monosodium glutamate. We also have 3 examples of basic amino acids. Basic amino acids are amino acids that are bases at physiological pH of 7.4. That includes histidine, lysine, and arginine. In all of those cases, they have a nitrogen or an amine group that has picked up, that has accepted a hydrogen ion, and therefore they have become an ammonium ion, and therefore they have a positive charge. Now what do we know? Why is this relevant? At physiological pH, and this is an important thing, this is an important preface, this is the list of polar, nonpolar, and polar charged, right? But this is at physiological pH. That's a critical detail. Because something that we haven't really talked about, even when we talked about acids and bases, we talked about how weak acids and weak bases had a difference in strength, right? The likelihood that they would give up that extra hydrogen or give up a hydrogen at all. Well, that leads to a concept that we didn't really talk about, which is the KEQ, or the equilibrium constant of dissociation. So in other words, what is the rate that is going to give up that hydrogen ion? That right there was a direct measure of the strength of those weak acids and weak bases that we talked about. Well, that measurement is dependent upon, not the KEQ, but the rate at which, whether or not an individual functional group is going to donate its hydrogen, is dependent upon the pH. So let me give you an example of that. These acidic groups right here, they are acidic at a pH of 7.4 because they donate their hydrogen. Is 7.4 basic or acidic? This is another one of those you don't have to remember everything from the previous chapters, except that you do, right? Because those are foundational concepts to help you understand the later chapters. A pH of 7.4, would you call that acidic or basic? Basic, right, which means that the hydronium ion concentration is relatively low. It is below 10 to the negative 7th, right? So, under those conditions where hydronium ions or hydrogen ions are relatively rare, then these two guys right here will donate their hydrogens to help out, to increase the hydronium concentration. But if we were to drop the concentration, if we were to increase the concentration of hydrogen ions, if we were to drop the pH into something very strongly acidic, guess what would happen? Now there's a whole bunch of hydrogen ions floating around. And hydrogen ions have an innate positive charge, right? And what do we know about positive and negative charges? They attract each other. And so if you have a whole bunch of positive charges floating around, what's the likelihood that one of them is going to find that negative oxygen and decide to be friends? Relatively high, right? So the likelihood of these amino acids to donate or accept hydrogen ions is directly dependent on the pH that that amino acid is currently at. Which means, and we're well above the slide where we talk about it, but it means that there are cases where this negative polar amino acid would suddenly become neutral, or would it just become polar neutral? What do you think? If we add a hydrogen to that oxygen right there, if I just put a hydrogen right here, would you call that for the side chain, look at just the side chain here, would you call that a negative polar or an acidic amino acid, or would you call it a polar amino acid, or would you call it a neutral amino acid? A non-polar. Does it become like these guys back here when we add that H? No, but it does become like these guys, right? It becomes kind of like that, or like that guy over there. It's still polar, right? Because we still have that electronegative bonds here. It's just not charged anymore. But if it's not charged anymore, that means it's not negative anymore. And what do we know about positive charges and things that don't have a charge. Do they have a particular reason to hang out with each other? No. Negative is attracted to positive, right? It's repelled from other negatives. Like repels like, right? And opposites attract. Sorry. But, breaking a little Paul Abdul there. But, negatives and neutrals, they don't care about each other. Positives and neutrals, they don't care about each other. So the reason why I'm spending so much time on this, we're going to talk about it in later slides, but remember when we talked about pH and why it was so important for biological systems, why you have the carbonic acid buffer in your blood to maintain that 7.4 pH? It's there because most of the enzymes in your body need that pH. We haven't talked exactly about why, but I've kind of hinted at it. The shape of those proteins gives it their function, and the shape of that protein is dependent on the things that hold it together that way, like positive-negative interactions. So if we change that pH and get rid of that negative, that might be getting rid of the glue that's holding that protein in that shape. And if it can't stay the same shape, it can't do the same job. And if you need that job to survive, then you're not in a good spot. So, acidic amino acids, by the way, if it's acidic or basic, it is obviously charged, but anything that is charged is also by definition what? Polar. It has an uneven charge across it. That's what polar means. Charged polar means we have a full charge. Instead of partial charges here and there, we've got a full negative or a full positive or even more than such. More than one positive, more than one negative. So what's that?
What about a molecule that has a charge on one side and it's neutral on the other? Still polar?
Still polar, because we have no charge to negative charge.
I thought when you were talking about hydrogen bonding and whatnot, it had to have a dipole.
Well, yes, but that's because a dipole is between two atoms. See, we don't have a dipole between one end of the molecule and the other. The dipole is the partial charge between, say, the oxygen and the hydrogen. The hydrogen is slightly positive, the oxygen is slightly negative. But that's still a polar molecule because there is a gradient of charge across it. But there's not a dipole between the methyl group on this end and the hydroxyl group over there. The dipole is between the two atoms that are giving you that partial charge. Or possibly the more than two, like a nitrogen and more than one hydrogen, that type of thing. But that's the dipole. So here we have our hydrogen acceptors at physiological pH. The only reason that they accept hydrogens right here is because there are enough hydrogens around for them to be attracted to it. If we raise the pH up to where it's much more basic, to where there aren't very many hydrogens around, guess what those guys will do? They'll let go of their extra friend and no longer be charged. So the pH of a solution directly impacts the charge of these charged amino acids that we are discussing. Specifically, histidine, lysine, arginine. We had another guy down here. Where'd he go? Here we go. Asparagine. See that guy right there? Glutamine, that guy right there, is that a charged group? No. Could you conceive of a pH where maybe it would be? Where we would have so many hydrogens around that maybe these guys would accept one and become an NH3?
Stomach acid?
In theory, at least, yes. As long as you can, in theory. This is why I'm pointing out, at pH, these guys are neutral. At pH 7.4, these guys are negative. These guys are positive. Now, a threonine, no, that's not fair. A leucine is never going to be charged. Doesn't matter what the pH is. Because there is no oxygen-hydrogen bond, there is no amine group in there anywhere. There's nothing that could gain or lose a hydrogen. So it's always going to be nonpolar. But those polar groups, they could conceivably become acidic or basic, depending upon the constituents of their side group. All right, Structural formulas. We call it the alpha carbon because that's the one that everything else is attached to. That central carbon, where you have the amine group hanging off one side, the carboxylic acid group hanging off the other side, a single hydrogen group as the third component, and then that fourth component is whatever the heck else that fourth component is, right? A hydrogen, a methyl, a propyl, isopropyl, isobutyl, whatever, right? Big ring structure that makes it tryptophan. We get a 3-letter or 1-letter abbreviation. All of those amino acids have that same orientation. What makes them different is that what was the orange group before, if I recall correctly from the slides, what is now highlighted in blue, the R group. So you can frequently see amino acids abbreviated with this exact same thing, but instead of actually drawing out the R group, they'll just put the letter R. When you get into an organic chemistry class, you get that a lot. When you're talking about organic chemistry class where what you have off to the sides of this doesn't matter, you'll frequently see R, sometimes even R1 and R2, meaning this is one side group, this is a different side group, but it doesn't matter what the side groups are. The chemistry at this location will happen the same way. So let's talk about the structure of how we combine these guys together. We have a special name. This is not the first special name that we have for a bond. The peptide bond is the special bond, the amide bond that forms between the amino side of one amino acid and the carboxylic acid side of another. This is just an amide functional group, right? But it's bonding the two amino acids together. But we don't just call it an amide, we call it the peptide bond. We call it that because it is a special type of amide bond that is ubiquitous in nature, i.e. all proteins put together in the standard way all share this exact same peptide bond between all of their amino acids. With a tiny little star that we're going to see how it breaks that rule a little bit later. When you first make the protein, they're all put together like this. We can then break it up into little chunks and cross-link it in different things, but when we're starting the process, it's always this. So we call it the peptide bond. Where else have we seen a special name for an otherwise common bond because it's freaking everywhere in that class of molecule? I'll give you a hint. We describe them all as either alpha or beta. What am I talking about? Alpha 1,4.
It was on the homework. I remember.
Yeah, it was on the homework. Alpha 1,4.
Oh, the saccharides?
No, it's as a part of polysaccharides, yes, but what was the name of the bond? Alpha 1,4, beta 1,4, beta, or alpha 1,6. Glycosidic. Close. Glycosidic bond. Alpha 1,4 glycosidic bond. The glycosidic bond, the bond between two different saccharides. Peptide bonds, the bond between two different amino acids. Question?
Okay, so I was just trying to backtrack what you said. So every protein bonded with an amino acid?
Well, proteins are by definition multiple amino acids.
Yeah.
So when you're building your Lego Death Star and putting those pieces together, they're being glued together via the peptide bond, which is a covalent amide bond. And it's another one of those things where we draw it like this, where it's a carbon double bonded to an oxygen with a nitrogen here. But remember, that nitrogen has that lone pair up here, right? Which means it's actually kind of more like this. So it's not really a 2 and a 1, it's kind of like two 1.5s. And because of the way that it is locked together, it allows for a limitation in rotation. If it was just a true single bond, then this could spin. But because of the nature of that bond, it is difficult for that to spin. That's a slightly advanced concept, but it will have some applications and better understanding of why proteins take the shape they do in just a little bit. When we take multiple amino acids and put them together, we call them peptides. So 2 peptides together are called dipeptides, 3 are tri, 4 tetra. And then much beyond that, we can keep numbering if we need to, but eventually we just call them poly. Like polysaccharides, polypeptides. In fact, an alternative name for a protein that's slightly more generic than protein is just simply a polypeptide. Because we have more than one amino acid put together with peptide bonds, polypeptide. So if we take glycine and alanine and put them together, we form an amine group. Do you see what happens? What's the process? This is a reaction that is creating a water molecule, so it would be a condensation reaction as opposed to hydrolysis. But if we can do it this way, it makes sense that we could also do it in the opposite way, right? In a hydrolysis reaction, which is exactly what happens when you eat a bit of steak, a bit of protein, or a bit of tofu. Your stomach acid is full of peptidases that will break those proteins down into smaller and smaller groups, possibly all the way down into individual amino acids so that your body can absorb them and use them as the building blocks for all of the proteins that you need to survive. So where's the water come from? They've color-coded it for you. 2 of those hydrogens are coming from the blue amine group, and 1 oxygen is coming from the pink carboxylic acid group. And so we form this amide, the peptide bond, and water. This can happen spontaneously, but for the most part this reaction is catalyzed by a very large structure called the ribosome. The active part of which, remember I meant 99.9%, the other 0.1 or otherwise, is something other than a protein. It's actually RNA in the ribosome that does the job of combining these 2 guys. It's RNA that does the work in the ribosome. So the thing that makes proteins is RNA, and then proteins do almost everything else. But the thing that makes the proteins is actually RNA. So here we have 2 different amino acids. Here's our glycine, here's our alanine. We're going to combine the 2 and pop off a water molecule. And here we get that peptide bond between our glycine and our alanine, giving us something that we can write out multiple ways, but we do so, the simplest way is to write it as glycoalanine. Alanine is the last one, the one that still has the carboxylic acid group on it. And then the thing that has the amine on it still, we start from the N side. Why would I call this the N side of this dipeptide? Because where do you still find the functional amine group? In this case, on what was the glycine, right? So that's the N terminal side of the peptide. This is the C terminal side of the peptide. Why? Because it's still got the carboxylic acid group, the full functional carboxylic acid group, right? So you start listing from the N by putting a YL, so glycine turns into glycol, and alanine is still there to react, so it's glycoalanine, otherwise abbreviated by GOY/ALA, or just GA. G for glycine, A for alanine. Here's everything I just described to you, but now with a tripeptide. So here we have alanine combined with glycine combined with serine in that order, and so we would name it alanyl glycosylserine. Now, does that mean that everything that combines alanine, glycine, and serine is going to be called alanyl glycosylserine? No, why not? Name me a different tripeptide using the same 3 amino acids. Somebody give it a try. Put them in a different order. let's just take serine and put it in the front. So that makes glycine the last one. So it's serine, alanine, glycine. What would we call that one? Seryl, alanyl, glycine. Right? Made out of the same three things, but in a different order.
Is that what we should recognize, or are you just saying we should pull them apart and stick them back together in a different order?
Well, it's because that's specifically what I asked you for. I said make a different amino acid, make a different tripeptide out of these three things.
Okay, but there wasn't one that we should be like recognizing.
No.
Okay.
And we have not talked about, at least not at length, I think we did have one tripeptide mentioned once as an example, something way earlier. But no, not asking you to recognize it. So, once again, this is the N-terminus. Why? Because it still has the amide group available to react to, right? This is the C-terminus. Why? Because it still has the carboxylic acid group available to react to. So if we wanted to add on amino acids, we could only do so at either the N or the C end, right? We couldn't add on amino acids, at least not via peptide bonds. We could conceivably, if we had a nifty enough enzyme, we could tack something onto that oxygen right there and start branching off right in the middle. But if we want to make a peptide bond, then we would need to add on to either the end or the beginning. The N or the C. And I will tell you this, that when it comes to biological synthesis, it's always the C. We always synthesize in the N to C direction. We're always adding on into them. The reason for that we may not understand until next chapter. All right, so that's amino acids, the different properties of amino acids, it's the different names for the amino acids, it's even how we combine them to form polypeptides via the peptide bond. So now, we're introduced to our list of essential amino acids. There are 9 of them. 9 out of 20 is kind of a large number. Though to be fair, we don't use all amino acids to the same proportions. But 9 out of 20 is still a, pretty high number. 9 of them you can't make. And therefore you have to get them from your food. Either directly from your food that already has them, or by feeding your microbiota and then they can make them for you, one of the two. But you can't make them. You have to get them from your food. This is why you can't just stop eating meat and replace it with carbs, because carbs is not going to give you the proteins that you needed, the amino acids that you were getting from that meat. You need to replace it with beans and other legumes. You need to replace it with things that actually have high protein content that can give you all 9 of these amino acids. Because otherwise you will start suffering from a dietary imbalance. You will start suffering from a lack of some of these amino acids, which might not mean immediate death, but can quite likely mean that you start having problems with things. There are disorders that were named for people who have an inability to appropriately digest or absorb some of these amino acids. For people who haven't eaten enough of them. We have diseases for things when you don't get enough of something before. Scurvy was from not getting enough vitamin C. Just eat a little bit of fruit and you'll be fine. Gout, goiter, not goiter, gout, goiter?
Goiter.
Goiter.
Not gout.
Thing, yeah, because gout's the urea in the joints.
Iodine.
Lack of iodine. We need some of these micronutrients, we need these essential amino acids. And if we don't have them, if we're short 1 or 2 of them, some of the enzymes that are critical for normal function will not be able to be made, and you will eventually notice their absence. Complete proteins, eggs, milk, meat, fish, beans, we've got protein, or peanuts and cashews and nuts down in here. Beans, black beans on the bottom.
Is it just humans that can't make those 9, or is that true of pretty much all life? Or all?
Different major groups of animals have different lists of essential amino acids. So different groups can make some. It's mammals can't, in the majority of the cases of those 9, but they can get them from the plants that they eat. And so we get them by eating the meat that they made from the things that they ate.
I guess Crichton in the first Jurassic Park, he engineered them so they couldn't produce lysine.
Right, he engineered them so that they had an additional essential amino acid that they would have to therefore get from their diet, which for something as big as a dinosaur, maybe you could say was okay, but plants make lysine too, so it's like that. But you could say, well, we have to give them extra or something like that, because normally there would be a synthesis pathway, and so you don't need to absorb nearly as much of it from your diet, maybe. It's a bit of a stretch. It works, but it's a bit of a stretch. It's not like he made that so that they couldn't make anything, and the only way they could get it was they made it themselves. All of the plants that they ate would have lysine in it.
Yeah.
But in the books, yeah, he just had to supplement it with lysine.
And that's how, everything gets resolved at the end, except for the hint that not everything is resolved. Mm-hmm. And it's because they start noticing that fields are being depleted of lysine carrying.
Right. The dinosaurs are figuring out which plants they need to eat to survive. They don't know why, but they're doing it.
Because they swim across.
So, primary structure. Now remember, we've been introduced to the idea of primary, secondary, and tertiary 2 times before. Where were they? Alcohols. Primary, secondary, and tertiary are alcohols. And what was the other one? Getting close. It was related to proteins, but we hadn't got there yet. Amines. Primary, secondary, tertiary amines. Amino acids. Give me a nice, there we go. Is this a primary, secondary, or tertiary amine?
Primary.
Primary. Because that amine is only connected to 1 carbon. Right? So amino acids are primary amines. At least the amino part of the amino acid is a primary amine.
It's the number of carbons it's attached to, or the ordinal number of carbon?
The number of carbon, in the case of amines, it's the number of carbons directly attached to the nitrogen.
Oh, okay.
And because of that lone pair, you can never have more than 3. You're not going to get a 4th carbon attached to the nitrogen because it already has that lone pair there. You can get a hydrogen come in and associate with it, but you're not going to get a carbon attached to replace it. But you can get 3 carbons to replace all 3 of the normal hydrogens. So that's a tertiary amine.
But the other one is its ordinal location, right? Whether it's the first carbon, second carbon, or third carbon?
Yes, so primary, secondary, tertiary, yeah, like first carbon, secondary, third carbon, 4th, 5th, 6th, whatever, like 3-methyl something. Okay. Yeah, that would be carbon number 1. I don't know if I'd call that primary, secondary, as much as just first carbon, second carbon, third carbon, but that nomenclature might be in some other organic textbooks that I'm just not familiar with.
Okay.
In the case of proteins, though, it doesn't have anything to do with how many carbons are attached. Because remember, amines is how many carbons are attached to the nitrogen, right? Alcohols was how many carbons are attached to the carbon that has the hydroxyl group. So we still couldn't get more than 3. Because, well, you've got to have the hydroxyl as one of the 4 on a carbon, right? So the other 3 might have been either hydrogens or something else, or carbons. Now, when we start talking about primary, secondary, tertiary structure of proteins, this is a totally different thing. Primary structure for protein is quite literally just how you spell it. So it is the sequence of the letters. So, bear in the woods. When I say that phrase, are you picturing a big hairy beast, or are you picturing a naked person?
The latter.
Which still might be a big hairy beast, depending upon which naked person you're describing. But The order of the letters, B-A-R-E, B-E-A-R, exactly the same letters, right? But a different order carries a different meaning. So, primary structure is the order of the amino acids. It tells you nothing other than simply the order, which means we could spell out a protein by just using one of the 20 letters that we use to abbreviate all of those things, right? We could just spell it out, G-A-C, just spelling it out. It might lead to a really, really long word that we could never in our lives pronounce, but we could spell it, right? So in this case, we have, okay, well, kind of, how many amino acids are combined into this polypeptide?
4?
4. How do you know that?
Because they put them at the bottom.
Right, they wrote them down there at the bottom. Without that, how could you tell?
They're peptide, there's 3 of them.
Right, how many peptide bonds do you see? How many amide bonds do you see in this chain of carbon? We've got 1 right here, which means 1 is here, 2 right there. Here's another peptide bond, so 3. Here's another peptide bond, so 4. What's missing right here? To make a 5th one, we'd need instead of a carboxylic acid group, we need an N right there and another peptide right here, right?
How'd you get the first one?
First one's right here,
Okay.
with the carboxylic acid group and the amine group. So our first peptide bond, let me draw them here. Here's our first peptide bond. Here's our second peptide bond. Here's our third peptide bond. So that means that we have 1, 2, 3, 4.
So it's always N-1.
Well, N-1 peptide bonds, yes.
Okay.
Or peptide bonds plus 1. Right?
Bookends.
You meant 4 peptide bonds.
No, 4 amino acids combined via 3 peptide bonds.
Or hyphens.
Or 3 hyphens, sure. No, how many peptide bonds? Count the hyphens, sure. That makes sense, everybody? I'll see it?
Don't even see it.
Ah!
Wait.
I'm drawing it again.
Okay.
I tell you, in the last class I took, I was forever snapshotting with my phone. Because she just, on to the next thing.
Yeah. I could see that. There was a class that I took, quantum chemistry, where you'd walk in the door, and there were 4 huge chalkboards. They weren't even whiteboards, they were chalkboards.
Already written out?
Yeah, you'd walk in, and everything was already written out. And he'd start over there, and just start going. And you'd get all the way to the end, and if he wasn't done, he'd go over there and start erasing. And adding to it as it went along. And so the first thing I started doing it, and then a whole bunch of people started copying. I came in with my phone and just took pictures of every board that was already on the board, so that when he started erasing it, I had that to go back to. I was still writing down all the notes and stuff, but I had the picture to go back to.
Way to go, Goodwill Hunting.
We tried it, I was like, we could turn these into PowerPoints for you. No, I don't need that. It's like, okay.
You sneak in and transpose stuff?
That was one of the coolest classes I ever took too, by the way, quantum chemistry. Just quantum nature and the Schrodinger's equation, just all of that. Atoms or atomic bonds as pendulums, just all sorts of awesome stuff.
But so once you have that going, doesn't it stop at that point being quantum because you're able to make observations without altering its state?
No, because any observation of the state does change the state. Because the only way to measure it is to bounce something off of it.
Okay.
The way you
can't
measure it
passively
is to
observe it.
change it.
You can't just passively observe that.
Right, because
Okay.
when I'm passively observing you, I'm not passively observing you. I am observing you as you were femtoseconds before, because I'm getting the light that bounced off of you. So you have actually moved past, the state of you that I see is actually you from the past. An imperceivably small amount of past, But that's kind of like that idea of when you look up and see starlight, there are stars out there that are dead. You are looking at ghosts. Because the light that they gave off hasn't gotten to us yet. We see another supernova go off. That means that the light from that star, from that supernova, has finally reached Earth. But that supernova actually exploded possibly hundreds of millions of years ago. It's been gone long before we were even around. But it just took that long for it to get to us.
You should trademark that unit of measurement.
It's kind of sad.
Femtosecond?
Oh, you said, oh, okay. I thought you were going,
No, femtoseconds? No.
So that hopes and dreams.
Yeah. Not all stars. Realistically, any star that's more than, what would it even be? Like 100 million light years away, arguably is dead. Because has died, possibly exploded, and new stars may have formed from it, and the light from those stars just hasn't gotten to us yet.
I don't think you can say
So
that
there's like
because
baby stars we haven't seen,
you can tell if you see a change in what's arriving,
like Charlotte's Web.
but
No, I'm talking about just our understanding of the life cycle of stars. Is that, and there might be some of the supermassive ones that live a really, 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 really long time.
Mhm.
That's possible. But for example, our sun has, or stars like our sun, has a lifetime of about 20 billion years before it's just going to go. And then it'll be effectively dead. It'll be a dwarf neutron star.
so I'm going to have to pay my student loans.
Yeah, eventually.
So like a couple
Yeah,
billion years.
5-ish billion years, yeah. If we manage to not nuke ourselves into oblivion in 5 billion years or so. And actually, a little bit before that, we'll be gone anyway, because the star will expand past Mars, I think it is, when it becomes a red giant, because it's running out of fuel and starts actually fusing hydrogen. Well, that doesn't create nearly as much energy, and so the contractive forces shrink, so it'll expand, and it'll expand past Earth's orbit, so the entire planet will be vaporized.
I'll be dead by then, so I don't have to worry.
That's like in 3 or 4 billion years, and then in 5 or so billion years, everything will go out, and all of the gases will leave off in a minor explosion, and we'll be left with a brown dwarf. So effectively just a big ball of iron that's just kind of slowly cooling off.
So it's gonna need to be a dwarf, it's not gonna remake itself.
No, the way, normally when you get stars that remake themselves is when you have these really big gas clouds, nebulas, where you get stars that form and then they go supernova again, and then that's kind of spreads the gas cloud out a bit again, but then it seeds it with other things as well, and then those stars will start to condense out of all of the gas that's there, and you get new stars that will then explode and reseed the gas cloud. And
Do black holes
That's
eventually
if you're
become
able to live in space. Yeah.
stars?
no,
Okay, I thought they.
a black hole is the terminal life stage of a life cycle of a star that's so large that when it reaches that point to explode, the explosion is actually an implosion where the gravitational pull on the outer layers is greater than the explosive force, and so it just shrinks in and gets more and more dense, which causes a runaway reaction that just further increases. The more dense it is, the greater the gravity for that mass, and so it just continues to compress down to an infinitesimally small point where all of the mass that was the star is now in that point. And then that creates this region of space around it where it bends back on itself, and so light can't even escape except for at certain points on the top and the bottom. And then that's what we call the event horizon. That's why it's black, is because any light that passes through that region can't ever get back out again, and will eventually cycle down into the center of the star. So black holes are black because nothing escapes them.
But they don't eventually suck in enough material?
To become a star? No.
Okay.
Because it all gets sucked down to that singularity. Like it doesn't make it bigger. It just adds more mass to the same volume of virtually zero. And so what that does is it increases the distance of the event horizon. The more mass it sucks in, the bigger the event horizon gets. But the volume of the star never increases. And so you can never kickstart fusion again. Because that's what happens when gas clouds eventually condense, is they get tight enough and small enough that the heat gets large enough that fusion kicks in, and then you get expansion into the star from the explosive energetic pressures of fusion versus the contractive pressures of gravity. And then for a while there you get burning until you run out of fuel, and then possibly boom, and possibly not.
You said on the event horizon, is there a gradient? Or there's time dilation?
As you approach it, there's going to be a small amount of time dilation. Yeah.
Okay.
But there's a point at which even light cannot escape. And as of yet, we haven't figured out how to go faster than light. So if light can't get away from there, then if you cross it, you're not getting away from there either. But that's astrophysics, not chemistry. So let's get back to chemistry. So primary structure of proteins is simply the order of those amino acids. Now you'll note, in this particular case, we have drawn the functional groups all up. This is R1, this is R2, this is R3, this is R4. Rotation around this bond is difficult. Not impossible, but difficult. I'm reminding you of this because we're about to see something where that plays a large role.
Then we're still erasing. What?
It's more circles, more boxes, more circles. Could you name? Well, they've already named them for you. Could you have named those? If I were to give you that right there and say, what's the third amino acid in this order? If, assuming, of course, that I didn't give you this thing down here in the list, would you be able to find it and name it?
No.
Is that just R or K?
R.
R.
It's
It's
R.
an R, okay.
The second one has a subscript of D2.
Okay, you can do this now.
Wait.
Just to correct the case.
RT.
Am I good now?
Wait. Sorry.
Like I said, I'm not in that much of a hurry.
Okay.
We have 3 days for 2 chapters.
Everything's right. Wait, so we're only doing, are we doing 15 and 16 and that's it, or?
15, 16, and 17. We've already done 15.
Okay.
This is 16.
Okay.
So you have 16 and 17 today, Tuesday, and next Thursday.
And so that's just R?
Yeah, just R1, okay, sorry.
R1, R2,
R-
R3, and R4.
4.
Thank you.
Right.
I mean,
No, I get it. But it's about, why is that one just R? Like, cause I just,
because
cause I,
sometimes it gets really complicated. They're like, well, I wrote it that way, so you should.
Yeah, I think it's good that your mind hasn't shut. I mean, I stopped taking notes because you're still trying. So more power to you.
hopefully I wouldn't ever pull something like that on you, but,
I mean, it's happened to me.
yeah.
So that's why
It's happened to me.
anxiety.
Happened to me. Tis better to ask. So, primary structure. Here they've done the same thing I just did, but they've color-coded it for you.
Yay.
So, thyroid hormone that stimulates the release of thyroxine is a tripeptide. This is not a protein yet because something we haven't seen shown here but is specifically notated in a book, which was honestly news to me, was that the protein classification starts when you have 50 amino acids. I didn't know that. That a protein starts at 50 plus. I don't know where that comes from. I kind of want to look it up. But it doesn't matter. So protein versus polypeptide. This is a tripeptide. 3 amino acids together. But this is the thyroid causes the release of thyroxine. So there are hormones. This would be a hormone, right? A chemical messenger that your body uses to communicate and cause other cells to do other things. Just like insulin causes your cells to take up sugar. Thyroxine is a tripeptide that causes your cells to respond in a specific manner. In fact, the release of thyroxine is caused by a thyroid hormone. So there are interactions. This is a hormone that is made by just combining 3 peptides. But it only happens if you do it in that order. Glu-his-pro, when you actually combine glutamine, histidine, and proline together, you get this hormone, Combine them in any other order, and you get a tripeptide that does nothing. Inject this tripeptide into someone's bloodstream, and their body will respond to it. Here's insulin. The first protein to be, I'm going to put sequenced in air quotes here. It was the first protein that was discovered where the sequence of the amino acids was discovered. It is a 51 amino acid long chain, 30 in a primary chain, 21 in a side chain, so 30 in this one, 21 in this one, where we have some cross linkages between some cysteines here. It's kind of hard to see because it's so small and thin. You could zoom in on it in your slides. But these disulfide bridges are between two cysteines because cysteine is an amino acid that has a functional thiol group, right? We have the same thing here between these two cysteines and then another cross linkage between these two cysteines, which seems like it would be awkward. Why would we get a cross linkage between two cysteines that are so far apart from each other on the same chain? And the obvious inference here is that, well, maybe it's not linear, right? Because linear, how do those two cysteines talk to each other? How do they stick to each other if it's all a straight line?
So there's a kink there?
There's more than one kink, and we'll get to that. But this is an example of, once again, the primary sequence of insulin. We have phenylalanine by valine, by arginine, by glucine, by histidine, by leucine, then cysteine, then glycine, then serine. So we have just the order of the amino acids, the way that it was synthesized together. That is the primary sequence of a protein. Right now, do we have... Yep. This guy right here, for a long time we used porcine insulin because it's very, very similar to human insulin. And so we would just isolate it from pigs because it was easier to get a lot of pig blood than it was to get people to donate human blood and isolate the insulin out of it. Now we can actually get this. We have bioengineered, I think the first step was they bioengineered pigs that actually made human insulin. And then the next step from that was they bioengineered yeast that would make human insulin. And that's even cheaper than pigs. So most of the insulin that is now medically available is made available because of our understanding of how proteins are made, the DNA that goes into the making and synthesis of them, and then the movement of those genes from one organism to another to allow us to make something in exactly the same thing. It's the exact same protein that comes out of this yeast that we purify out of the yeast cultures. But it's a heck of a lot cheaper to make en masse than when we were growing it in pigs. Or even just getting it straight from pigs.
Franken yeast?
Was that?
Franken yeast?
Franken yeast, yeah. I mean, it's definitely a genetically modified organism. It's a GMO. But there's a lot more GMOs than most people realize. Modern society wouldn't have been able to advance to where we have without it. Whether or not we're doing so responsibly is a different discussion, but... All right. There's another group, enkephalins and endorphins, natural painkillers that you guys are probably aware of. Endorphins, always like, hey, you need to get that endorphin rush. Enkephalins are found in the thymus and the spinal cord. They are pentapeptides, the smallest molecule with an opiate activity. They bind to opiate receptors as painkillers. Glutamate, glycine, lysine, tyrosine, alanine, asparagine, lysine, isoleucine, alanine, aspartic acid, lysine, phenylalanine, and leucine again. Beta endorphin. Take those exact same peptides, put them in a different order, and you get a peptide. In this case, a nonapeptide, a pentapeptide, 10 peptides, 10 amino acids. But it would do nothing. But those 10 peptides put together in that order gives us a molecule that you have receptors for that causes a relaxation of pain, a release of pain.
So is that like the start of a side chain, or did they just run out of room?
I think they just ran out of, well, phenylalanine kinks.
Okay.
So it's a thing where the side chain actually links back to the amino acid chain, and so it causes kinks in protein. They might be showing you that, or it might just be, yeah, they ran out of room with the image on the like,
St.
text
John's
box.
Hospital.
St. John's Hospital?
St. God's Hospital. Oh, I thought you mentioned idiocracy at one point.
No. Well, a few slides back, but not today. There are two hormones that come out of the pituitary gland. You might be familiar with some of these, oxytocin and vasopressin. Hmm?
No, it looks just a little different.
Yeah, down here. They're exactly the same thing, where we have a cysteine, tyrosine, and then right here, oxytocin has an isoleucine and vasopressin has a phenylalanine. And we have glycine, asparagine, cysteine again with that disulfide bridge, so linking the two cysteines, another proline. Then we have leucine and arginine, so oxytocin has leucine, vasopressin has arginine, and then they both end in glycine. So these guys are exactly the same except for the protein in location 3 and 8. Oxytocin stimulates uterine contractions in labor. So if you make this protein with an isoleucine and a leucine right there, then you get a hormone that if you inject it into a woman that is currently pregnant, actually that might work even if you're not pregnant and just cause some uterine pain, but it's what we use to induce labor. My wife had to be dosed with this twice
Yeah.
for both of our children.
Sucks.
I thought they used Pitocin for that, because oxytocin has other uses.
It does, but Pitocin is a generic and synthetic version of oxytocin.
And when you don't have pain meds, it really sucks.
Right.
Trust me. Oh my God, horrible.
So then we also have, if you put phenylalanine and phenylalanine and arginine in there, then you get vasopressin, and that's an antidiuretic hormone that adjusts your blood pressure by changing how much your kidneys reabsorb water. So, depending upon which condition you're trying to treat, that sequence might be really important.
So is Cervidil, I don't know if you know what that is. I was curious if it contains the same thing. They use it to ripen your cervix. It's like a dissolvable pill. I was just curious.
I would assume not, because the flexality, the stretchability, and how much your cervix is actually expanded, that's a very different thing than the contractions.
Well, it's used in like the exact same way. I was just curious if it works in the same way.
I thought they used ethylene for that.
I don't know.
No, I'll double check.
Seriously? Ethylene?
To ripen.
Yeah.
It's not an apple, a bushel or otherwise.
You got that from the tomatoes, didn't you?
I got from earlier lectures. So that's
Yeah,
why
the
it's-
tomatoes.
Ripening the tomatoes and apples, yes.
The whole thing will work like that.
Nice reference. Took me a second. I'll admit it. Point is that the order of the amino acids is really important. Different amino acids are going to produce different, even the same amino acids in a different order is going to produce a different molecule that will therefore have different biological effects. So, we now have a good understanding of primary structure, right? Primary structure is just the sequence. There's not a lot of structure in the primary structure, but we call it the primary structure because when we're talking about it in terms of proteins, the reason we're talking about it is because it determines the structure of a protein. The reason that these guys have a different effect is because when you change those two amino acids, you create a slightly different structure. And so we can continue to describe it, since it's the primary structure, that would imply that there is more than one, right? Otherwise we would just call it the structure. So the secondary and tertiary and quaternary structures are what comes next. And it's going to have a lot to do, as it's mentioning right here, with hydrogen bonds between the carbonyl group that's still left over in one amide group, and the NH group from the next, as it says here, the next turn, which is an interesting thing to say, but we'll get to it. So, these are alpha helixes and these are beta sheets, or at least in the way that they are most frequently drawn when we're trying to give us a pseudo three-dimensional appreciation for the structure of an overall protein. Here's our alpha helix. In an alpha helix, you get this series of amino acids, but they bend and twist back on each other, and then that structure is stabilized by hydrogen bonds between one amino acid and another amino acid that is a certain number of steps forward. So it's like rungs in a ladder, sort of, because ladders work horizontally instead of vertically. But what we're taking is that series of amino acids and we're twisting it into a spiral. But when you do that, what that does is it shoves the carbonyl group of that amide from one amino acid out and orientates it just over the hydrogen coming off of that amide group. And so we get the positive from the hydrogen associating with the negative from the oxygen, and that gives us an oxygen-hydrogen bond that we call a hydrogen bond because it involves the partial charges of a hydrogen. And that stabilizes that twisted helical structure. You'll note that we are not drawing the R groups here. In fact, the R groups are being illustrated by one ubiquitous dark blue ball. Now those R groups are sticking off to the side, heading out this way in this guy, that way, and then going in the back on that one there, coming kind of out that way here, coming kind of out this way here, out that way again, and then going around to the back again that way. What are those R groups? Anything we've discussed so far, sort of. You can put a little star next to anything because of something we're going to mention in a minute. But the point is that this is a corkscrew, an alpha helix. It's stabilized by hydrogen bonds on the backbone. What do I mean by the backbone? Forgive me, going back a few slides here. This is the backbone. CCN, CCN, CC, there was another one, N. That backbone of the amino acid chain, amino to acid to amino to acid to amino to acid to amino to acid, that's the backbone of a polypeptide. The side chains don't matter so much. And actually, this is probably because it did just run out of room because that's a phenylalanine, not a proline. Proline's the guy that causes the cake because of that link, that circular link right there.
So it really,
Oh, yeah. For mediocrity, yeah. Ran out of room. All right, so here's an alpha helix. Here's a beta sheet. How is it different? A beta sheet has exactly the same thing. We have this, the amino to carboxyl, amino to carboxyl, amino to carboxyl, amino to carboxyl, right? Except now instead of having all the R groups pointed out from the center of the corkscrew, now we have them pointed up or down here, up here, down here, up here, down here. We have a bend here to imply that there's other guys out here. We don't just break it. There are other amino acids. In that gray area there that they're glossing over. That's a 1, 2, and N dot dot dot, in case you can't see what my shorthand is. I don't want you to think that it's literally just snapping off and forming a beta sheet. That's not what's happening. But a beta sheet is the other shape for secondary structure. Here's a clue. Secondary structures has two shapes, either the alpha helix or the beta sheet. There are two possibilities for secondary structure, which is also kind of lying. There's a third possibility of bit that's just kind of hanging out there, like this gray bit, right? But if we're going to name it, there are two possibilities for secondary structure, either alpha helix or beta sheet. What's holding this beta sheet in the shape that it is?
Hydrogen bond.
Hydrogen bonds. Between what?
Can't quite read it.
Hydrogen bonds between peptide backbone. So it's exactly the same thing that held together the alpha helix. So why is this a beta sheet and not an alpha helix? It would be a valid question, and the answer is, well, it depends on what those R groups are. You can take the exact same 10 amino acids, and if you order them in a certain way, they will naturally twist into a beta sheet. Twist into a beta sheet, twist into an alpha helix, forgive me. They will naturally twist into the alpha helix, the corkscrew, held together by hydrogen bonds.
Without water?
In water.
Okay.
In an aqueous solution. You could take exactly the same 10 amino acids but put them in a different order. And now they will form a beta sheet, still held together by hydrogen bonds in the backbone, but taking a different shape. So, what determines the secondary structure?
The location of the R.
The location of the R groups? Sure. And how can you tell? What would you use to describe the location of the R groups? What have we already described that tells us the order of the R groups?
Chirality?
Not chirality, but the primary structure, right? It's the order of the amino acids. That tells you the order of the R groups, because each amino acid has a different name, different abbreviation, that tells you what that side group is. The primary structure determines the secondary structure.
Will the pleated sheets, will they Pringle?
But no, they won't compress. No.
No, I just, like, layer.
They can, yeah.
Okay.
Depending upon the orientation of the R groups. There is another aspect for secondary structure that doesn't apply universally, but is still a manner of describing a secondary structure, and that's the triple helix. And that's 3 polypeptide chains woven together. And we see this primarily, at least with this example, in collagen. Collagen is a very important connective tissue that you find all over the place. Tissue, skin, tendons, cartilage. It's 3 peptide chains wrapped around each other and held together via hydrogen bonds of the backbones.
So how did it go from a twisted to a braid?
I don't know if it's really braided, woven together.
The crossings are, so.
Right, I mean, here it's shown as a cross. I don't know if that's quite literally the way it's supposed to be, as much as they just wrap around each other
Okay.
and hide. But either way, this formation is assisted by other peptides. It isn't so much spontaneous association, it's proteins that are built and put together to have that final form.
Gotcha.
So it could be braided because of a way a protein organizes it as it's putting it together. Alzheimer's disease. We're familiar with this, right? Actually, before we get into Alzheimer's disease, let's take a break, stretch our legs. 7:10, we're halfway done. And we're halfway-ish done with this chapter. So,
This is the same as spongiform encephalitis and BCE? Mad cow?
I don't think so. But I cannot say so from an informed position. I don't think so.
Coffee.
Did you say I'm awful?
I want that.
No, that I'm awful? I thought I heard somebody go, he's awful.
You're awful.
Pretty sure that's what I heard.
If I wanted to call you awful, I'd say it to your face.
Don't fight it. Don't fight the urge.
We'll just throw things at you.
So who all in this group is going on to physiology? Or, and then nursing, 209?
Me.
There's one other person I know for sure. Are you?
I don't think it's me. I don't have bio, a math class, and something else.
But what are you hoping to apply to nursing school?
Oh, I take the test this next upcoming semester, so I should be in
Spring?
the spring, yeah.
So
So it's-
you could be in the same cohort. That'd be awesome.
I'm taking the test in August, and then if I want to retake it, I'm taking it in October.
Yeah, I'm taking T's on the 23rd.
Of
No,
August.
I'm
Oh, I had a friend and she wanted me to take it with her. She's like, I'll pay for you to take it with me.
Majors and non-majors, so most people.
Well, yeah, but I mean, I didn't know if like everybody was, and she's clearly not, so, you know what I mean?
She's not what?
She's not going for help.
Oh,
I'm criminal justice.
you're what?
Criminal justice. I have to take this class though.
Criminal.
You don't have to take 110?
Criminal justice.
Oh,
No, I
criminal
have to take
justice.
110, but I have to take this class too.
You take 109 and 110? Really? Boy, it's changed since, I have a minor at that, but
I have a
minor
minor
in
in just the
for-
criminal part.
there's a minor in criminal justice
I'm doing my
forensics.
minor in law. Criminal law.
So nursing degree with a minor in criminal law?
I've always been an argumental person.
No.
Me too.
And she just said while you're gone that she's confrontational. Sorry, I'm like super fatigued and my mind's all over the
I'm
place.
just
I'm
gonna
trying to
work
keep my
with
mouth
dead
shut
people.
today.
I'm just gonna work with dead people so they can't talk back.
I had a friend who went into vet school
Mhm.
specifically for that. He was like, I need a job that pays well, but doesn't force me to interact with a lot of people.
That's why I'm working with
I
dead
want to be a
people.
veterinary pathologist. So I'll work with animals, but they'll all be dead.
Yep. That's what I'm doing. I'm working with dead people.
Got to do a post-mortem on a 15-foot boa constrictor.
That's
That'd be interesting.
And the funny thing is when we started the dorsal cut, it's like the snake exploded because they had IV'd in so much antibiotics
Ew.
that I'm surprised it didn't drown from. Kelly wasn't here,
Yeah.
and that I don't think the young lady sits all the way in the back by the door.
Amber wasn't here,
Excuse me.
yeah. No. Megan. No, who am I looking for here?
Are you Amber then?
Yeah, Megan. Megan.
She's not all here. Wait, no.
Megan's the one that hasn't been here a lot.
I don't see how you could not come to this class.
I know. See, this is the thing. All right, so this is the problem. There's only 11 of you guys, and only about 8 of you show up regularly. But I'm horrible with names,
Me?
and I need to be able to associate names with faces. And one of the things that CC doesn't do
Put a
is
picture.
yes, it does. Now, a few of you have
It's
emailed
not
me.
on the email thing. If we put it there, it's
Right,
Yeah.
if you put it there and you email me, then I can see it. But if I go to click on your
Of course,
name
mine's all
anywhere in
one
D2L, it does not show me
Yeah.
your picture.
I don't.
So
look
yes, a
like
few of you have
I'm
sent me emails.
So if
When
you
I
go
see
to the
email, I
discussion
can see
board
it.
and people have contributed,
Yeah,
and
you
they
see
put it on
there.
their profile.
Right. So there's a lot, but see, back at MS&T in Mizzou, the ID picture that you get taken, that's
Mhm.
automatically associated with your student profile. And so any class that I ever teach, I could with like 4 clicks print out a list of names and pictures. And so I could associate, and like for my lab classes, what I do is I take pictures and names and I'd make myself a cheat sheet for these 4 people are at that table and these 4 people are at that table. And so that way, by about halfway through the class, a little less, I'd know everybody's names because I'd have to like reference who was missing from each one because it was group work, so I had to know.
What learning environment at Mizzou?
No, this is down in MS&T, down in Rolla.
Oh, okay.
They used to do that at Folsom Lake too.
What's that?
They used to do that at Folsom Lake too.
Yeah, yeah. Is it and I can kind of understand why they don't, because there's all like privacy of students maybe, but at the same time it's like it would really benefit. So outside of me like going, all right everybody, let me get a picture of you at the back of class on the first day, like I don't know.
You're from
Or just
California?
awkwardly like doing a roll call and then awkwardly staring at you every time I call your names. Like I'm trying to, this is not me being freaky, this is me trying to memorize your face.
Amber, you're from California?
Mhm.
So like Marin County, Sacramento area, or what?
Sacramento area.
Oh, cool.
I used to live in Placerville,
Oh, no.
California.
I can't imagine why you
Maybe
left. Huh?
Do you know where Pollock Pines is?
Say it again?
Pollock Pines?
Uh,
No? Do you know where Pollock Pines is? You said you used to live in Placerville.
No, I lived there like, let's see, how old is my daughter? 25 years ago. So I don't remember nothing.
1989.
It's next to
I
Plattsburgh.
lived there in
It's like 30 minutes away
93.
from there.
I did my first 11, 12 years in Los Angeles, then we moved to Mariposa for a year. So we own Yosemite. We let a lot of Chinese people use it though. It's so weird. You go to town and you get a haircut, and you're sitting there in the thing. You're getting a haircut, and one of the buses would come through, and suddenly all these little faces are pressed up to the glass, watching the Americans get haircuts.
Okay, you find that a lot of times in LA and stuff like that, there'll be like Chinese tour groups going through LA.
Oh really?
They'll just watch you. I'm like, okay. It's a whole bunch of other, like once people visit the United States, I like to go to California more because, it's wonderful. It's a wonderful state.
So Kelly, Megan,
Oh please.
Mary,
I hate
who else are we missing? We're missing one more.
The Golden State. Or the,
What is,
Eureka.
no, she usually sits either there or here. What's her first
What were
name?
you saying?
I don't remember.
Oh, the Gold Rush State, right? Okay.
Eureka. I found gold.
Eureka!
Let's not comment on people's hygiene here.
And plus, we have Hollywood stars, so people like going to Hollywood.
I was born in Hollywood. I was born in the red light portion of Sunset Boulevard. Because that's where Kaiser Permanente is. And Mom worked there as a pediatric oncologist. No, I'm sorry. Pediatric oncology nurse. We should have been living a lot better if she was an oncologist. So how Missouri, how?
Well, my mom, their company was closing down. They
Mhm.
got bought out, so they drove a couple of us from California over here. So
Aerospace?
Huh?
Was it aerospace?
No, it was ABC Labs.
Oh, okay.
It's called something else now. They changed like a couple names. But yeah, it was originally ABC Labs.